hello everybody. Now let us continue our discussion further on co-crystals. Today we will be discussing on pharmaceutical co-crystals, because the primary applications of co-crystals is in the field of pharmaceuticals, where it has been observed that drugs which have got a very poor solubility profile can be now chemically modified so as to improve the physicochemical properties of the drug. And this has got a lot of applications in the pharmaceutical industry, because the manufacturing of large amount of drugs where the different physical and chemical properties have been tuned depends upon this process of crystallization. So in the process of pharmaceutical crystallization, the drug which is also referred to as the API, that is the active pharmaceutical ingredient, is an integral part of the pharmaceutical co-crystal, and the co-crystal is a two component crystal, which is a unique phase of the substance, which consists of the drug and another substance which is called the coformer. And the coformer is a component which must be soluble in water preferably, and must also be non-toxic. So these kind of substances which are non-toxic and which are chemically benign are referred to as GRAS, that is generally regarded as safe. And as we have already looked at the classification of co-crystals, that it can form either co-crystal, it can form a salt, it can form also solvates and polymers of co-crystals. So there exists diversity in the co-crystals as well, and it is also important to keep in mind that crystalline drugs are important, but we also have the amorphous drugs which are characterized by high value of the Gibbs free energy, and therefore the dissolving property of these amorphous forms is extremely high. But the problem with the amorphous drugs is their intrinsic stability, and these have a tendency to convert to the metastable form and finally into the thermodynamic form. So industry would always like to work with amorphous forms which are more active and having characterized by high values of the Gibbs free energy, because they have higher solubility and dissolution profiles, but there are issues associated with its stability. Also, it has to be kept in mind that solvates, particularly hydrates, are also useful because they have higher solubility properties, but if they tend to lose the water molecules, the solvate structure can collapse, and that can lead to the loss of crystallinity of the drug, keeping in mind that the crystalline form is the more active form. Now, why this field is of extreme relevance in terms of application in the industry? Because it finds application in the design of patents. So patent is where you have an invention and you would like to protect that particular invention. Now when you say that research related to pharmaceutical ingredients can find their way in patents, because you are increasing the innovativeness of the process, you are enhancing the property, and in this regard a patent becomes a viable option. But before you think about making your research in pharmaceutical co-crystals into a patent, you have to think about the novelty aspect, the non-obviousness of the process, and the utility of the process. So novelty, it should not be something which is very obvious, and it should have applications or utility to serve society. So in terms of the novelty, you have got a new composition of matter. Okay, so novelty is that you have got a new solid state formulation of the drug. Non-obviousness is because in this case the identification of the co-former is important, and it is never a routine thing. So identification of the co-former which is going to bind with the drug to form the co-crystal is never routine. Unlike the case of a salt wherein an acid combines with a base to give you the salt and water. So that is actually the reason why this process is non-obvious. The novelty is because you have a new solid state formulation of the drug, and the novelty also lies in the non-obviousness lies in the process that the design of the crystal requires logical thinking, reasoning, and knowledge of intermolecular interactions. So based on these constructs, you should be able to design new co-crystals with new applications. And the utility part is, as I told you, the enhancement in the physicochemical properties of drugs like dissolution, solubility, and these have implications in bioavailability of the drug. So so now we see that co-crystals affect a large number of properties with respect to the parent drug. Just to reiterate once more, some of the most important properties which are of relevance, keeping in mind pharmaceutical co-crystals and the impact in the design of new drugs and enhanced performance compared to the parent drug, is the solubility, the stability, the hydration properties, the melting point, the mechanical properties like flowability and tabletability, and finally the PK, that is, I mentioned the pharmacokinetics, the pharmacokinetics profile like bioavailability and permeability. And just to give you an example, for example, the drug, there are some drugs which are actually liquid at room temperature or at temperatures between say 0 to 20 degrees centigrade, and the idea is to improve the stability of this drug. One such example is propofol, which has got a melting point of 18 degrees centigrade, and a co-crystal has been formed with isonicotinamide plus propofol, and the melting point now is 55 degrees centigrade, and thus this exhibit enhanced physicochemical properties. So this is just to give you an example that how co-crystal can actually stabilize a drug which is actually quite active but exists in the liquid state almost at near room temperature, like above 20 degrees centigrade it exists in the liquid state, but it can now be stabilized and the formulation can be made into a solid by co-crystallization with isonicotinamide. So keeping in mind the fact that drugs have to be developed, the pharmaceutical industry have classified the BCS system, which is the biopharmaceutical classification system, where we have now effectively 4 domains which are characterized by high permeability and high solubility, high permeability, low solubility, low permeability, low solubility, and then we have got high solubility, low permeability. So this is called the BCS1 classification. This is the BCS2, this is the BCS3, and this is the BCS4 classification. So those drugs which are actually available in the market belong to the biopharmaceutical classification 1, which are characterized by high permeability, and they are also characterized by high magnitude of the solubility. In other words, this is the increasing solubility range, and this is the increasing permeability range. So in the second one, you will be still in the high permeability region, but there will be a drop in solubility. When you come into this particular region, it is the BCS class 4 drugs, and this is the BCS class 3 drugs. So the idea is to improve the classification. If you have a sparingly or a drug which has got poor solubility, for example BCS class 2, or you have got a BCS class 4, then you would like to improve the solubility and go to the BCS class 1. Or if you have got a low permeability, you would like to go from BCS class 3 to BCS class 1. And what has been observed is that many active drugs which enter into the pipeline of clinical trials at the later stage have been observed to have issues related to the solubility and permeability. And the idea is now to employ the method of co-crystallization to be able to improve these physicochemical parameters. And it is also important to keep in mind that what kind of drugs are being made, because drugs have got a hydrophobic part and they've got a hydrophilic part. So those drugs which are mostly hydrophobic, they tend to pass the lipid bilayers. They have to be transported across the lipid bilayers of the cells, whereas those which have got hydrophilic groups are the ones which stay in the blood serum. So that decides the mode of transport of the drug. And it is also important to keep in mind that permeability is a very important factor, because the drug has to be absorbed across the gastrointestinal tract. So permeability is very important, and permeability can be modified by the modification of the structural features. By the modification of the structural features of the drug, now you can actually improve the modification of the structural features can improve the problems of absorption and distribution of the drug across the gastrointestinal tract as well. So permeability also is another very important factor. So we need to actually now design drugs using the technique of co-crystallization, which have got enhanced solubility, and so that we can work on the bioavailability of the API or the drug as well. Now the concept is that once the drug goes into solution, or it goes into the blood serum, it should be available for a sufficient period of time. There are some drugs which perform the necessary action, and in a short period of time the drug actually crystallizes out or precipitates out and is then metabolized and excreted out of the system. But there are some diseases where we need a sustained release of the drug into the blood serum, and which is available to the patient over a sufficient period of time. So in that process, we need to maintain the required available concentration, which can exhibit or exert the necessary pharmacokinetic effects. So this in order to look at this process, that is the understanding of the pharmacokinetic profile, we can now look at this pharmacokinetic profile as follows. So we have the concentration in plasma in microgram per ml as a function of time, which is given in hours, and essentially this profile looks like this. Okay, and here we can say this is 0, 5, 10, 15, 20, 25, and then we can have 2, 4, 6, 8, 10, 12, 14 microgram per ml. This is the y-axis, and the x-axis is the time. And what we can now represent here is, this is the Cmax concentration. This is referred to as Cmax, and this is the corresponding Tmax. This particular region, starting from 0 till the time the drug reaches the maximum concentration, is the process of absorption of the drug. And what is essentially important is to probe this entire process for a period of 1 day, that is 24 hours. So it essentially tapers off to almost a value close to 0, and after the Tmax it follows the process of elimination. So the elimination process of the drug also starts, although we do still have some features of absorption, but mostly it is the elimination which is important. So this is the maximum concentration which we have, and this is the minimum effective concentration, and in this is the pharmacokinetic parameter, that is the therapeutic index, which we of the pharmacokinetics profile. So this is the therapeutic index. So so we can see that Cmax represents the maximum concentration, Tmax represents the time necessary to achieve the release of the drug to achieve the maximum concentration, and the total area under the curve, is represented as AUC, which actually signifies the dosage of the drug, Now, to start with, you can know the solubility profile of your drug or your co-crystal, and co-crystal is supposed to enhance the solubility profile, but that solubility profile might still not reflect the dissolution profiles which is necessary to achieve the necessary therapeutic values. Okay, so in that regard, we have to make sure that the available concentration can exert the necessary therapeutic value, and that has to be achieved keeping in mind that the equilibrium solubility of the drug or the co-crystalline solution might not have sufficiently high values for it to exhibit the necessary therapeutic index. So in order to be able to do that, and as we said, we have now made salt co-crystals. We can have a salt where the solubility can be enhanced, and how this apparent solubility is achieved, enhancement in the apparent solubility is achieved for these poorly soluble drugs can be explained by the concept of spring and parachute model. And what happens in a spring and parachute model is as follows, that we now have so in a spring and parachute model, what we can now look at the spring and parachute model, which is necessary to understand the solubility profiles of a drug in a co-crystal as a function of time. So we can now look at this spring and parachute model. We have the time axis here, we have the drug concentration. A drug concentration can be done both in vitro, that is in glass, as well as in cells, live cells, that is in vivo as well. So to start with, we have this increase in dissolution, and then there is the thermodynamic equilibrium which is achieved, where the solubility essentially becomes a constant. Compared to that, we have got the spring effect, and then what we actually need is, so this we can say is like the parachute dropping here, and here we can see it is like a spring. So when you release the spring, there is a sudden spring motion is like when you have actually is when you have compressed the spring, it stores the potential energy, and now when you release it, there is a displacement, and that release of energy is now converted into kinetic energy. So the spring motion essentially symbolizes the fact that you have a capsule where you have kept the drug compact, and now that drug is dissolved and it is released into the solution. And you can see that there are three processes happening here. The first, which we said, is the simple achievement of the equilibrium solubility. The second one is only the spring effect, and the third one is the spring and the parachute model. And so to start with, if you have in the process 1, you have a crystalline form of the drug. You have the crystalline form of the drug which has actually been co-crystallized with your particular co-former. But to start with, we have the crystalline form, and that has got a low solubility. And then in case of 2, we have a short-lived metastable species. A short-lived metastable species which achieves the Cmax value within a period of half to 1 hour. It achieves the Cmax value, and then it converts within a short span of time into the low solubility crystalline form. So the crystalline form, which is the thermodynamically most stable form and which is less soluble, the short-lived metastable species converts into the low solubility crystalline form. That means the one which is getting released into the solution is essentially an amorphous form, and because the amorphous form is not very stable, it tends to convert immediately over a short period of time, say between half to 1 hour, into the corresponding crystalline form. In the third case, so this is the spring effect, and then you have got the parachute effect, where you have got now a highly soluble drug form which has been actually co-crystallized with a suitable co-former, which is a metastable form and which is stabilized by the co-former. Due to hydrogen bonding interactions, and it is and this particular association between the drug and the co-former is what maintains the necessary solubility profile, and it is only very slowly that the drug is being released, and this particular metastable species converts into the low solubility form after a much longer time. So to start with, we have a highly soluble drug now because it's co-crystal. It dissolves into the biological medium. It maintains the necessary concentration of the drug in the blood plasma over an extended period of time. Say the time is now extended from 1 or 2 hours to at least 4 to 5 hours, and that is the parachute effect. And then finally it goes down to the equilibrium value of the equilibrium solubility, where it has converted into the more crystalline form. And we can look at this entire process physically as follows. So we have, for example, a particular form of the drug, okay, and we have co-crystallized it with, say, a co-former. So we have the co-former here, and this is the crystalline form, and then the co-crystal dissolves. So once the co-crystal dissolves, we have these random aggregates now. So we have these random aggregates, so they constitute the amorphous phase because the crystallinity is lost, and this is the spring effect, where you have an active amount of these drug molecules floating around along with the co-former molecules. So this is the amorphous phase, and now this amorphous phase can either convert back. So before you formed this particular co-crystal, you also had the parent drug. So it can actually convert from the amorphous phase into the low solubility crystalline form, or it can also convert into a metastable phase where the arrangements now are different. So you can have a different arrangement now of the drug molecules, and the co-former molecules can be sitting here, or sometimes it is not necessarily the co-crystal forms, it is simply the metastable form which forms. So we now have a metastable form, and which now exhibits the parachute effect. So we start with a drug on a particular crystalline form, make the co-crystals which has got enhanced solubility, dissolve it, and it makes an amorphous phase. The amorphous phase creates a short-lived species which can increase the drug concentration now and then get converted into the more crystalline soluble form, or it can then convert into the metastable form, which essentially exhibits the parachute effect. So these kind of processes takes place, and this very nicely explains the enhanced solubility in poorly soluble drugs. So keeping this in mind, let us look at one particular example where we can look at co-crystals in the drug riluzole. Now riluzole is, you can see this is the structure of riluzole. It contains a CH3 group and it contains an amino group here, and we have tried to co-crystallize this with different diacids like malonic acid, succinic acid, glutaric acid, adipic acid, sorbic acid, and also some bases like dimethylaminopyridine and nicotinic acid. And this compound is actually used for a treatment of amyotrophic lateral sclerosis, and it is a highly water insoluble drug at neutral pH. And the idea was to make new co-crystals and improve the physicochemical properties of this particular riluzole drug. And we got many co-crystals, and then we also got for adipic acid we got two particular forms, form 1 and form 2. We got co-crystal with malonic acid, succinic acid, then we got it with pimelic acid, then we got it with azelaic acid, dimethylaminopyridine, and we also got it with nicotinic acid. And you can see that the melting point of the co-crystal is in between the melting is basically close to the melting point of the drug, and that is very interesting because you don't essentially change the melting point to very high values, otherwise the lattice energy also is higher, and that has to be overcome when the co-crystal dissolves in the biological medium. So the melting points of the co-former are given here, and in most of the cases the melting point of the co-crystal is comparable, like 114, closer to that of the riluzole itself. In case of sorbite it is lowered, the melting point is even lowered, whereas in case of SBRA it is actually enhanced to 136 degrees centigrade. So keeping this in mind, now I can show you some of the interesting structures which have been obtained by co-crystallization. So here you can see this is the adipic acid which contains the CO2H, CH2, 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 CO2H. It is held by the strong NHO and OH and hydrogen bonds. This is the most important hydrogen bonding which connects the co-former with the drug molecule, and then the drug molecules are further connected by the weak CHO and CHF hydrogen bonds between the riluzole molecules, and this forms a layered structure, as you can see. So this is the structure of the co-crystal phase. Then we can see with malonic acid as well, and in the case of malonic acid you will see that it has formed a salt. So salt means that one of the carboxylate hydrogens, this particular carboxylate group, has lost its hydrogen and it has now protonated the corresponding nitrogen of the riluzole ring. So now the nitrogen gets protonated as, and it forms N plus H. So this particular nitrogen here now gets protonated. So you have the salt formation, and again you can see the arrangement of the hydrogen bond. This is the NH O minus, and this is also this is the neutral NH, and this is the protonated NH plus O minus. So you get these subtle changes in the hydrogen bonds, one having the charge species and the between the NH and the O minus. And so you have this kind of a co-crystal structure. In the case of nicotinic acid, it is interesting to keep in mind that nicotinic acid itself exists as deuterion. So you have CO2 minus here and the N plus H, this is the deuterion, and then this forms hydrogen bonds with your drug molecules. You can see here the hydrogen bond. This is the NHO hydrogen bond, this is the CHO hydrogen bond, and you have this network of hydrogen bonds which connect the drug molecule with your co-former, that is the nicotinic acid. So these are the physicochemical properties. So you can see that the dissolution rate has been enhanced for your for sorba, that is your sorbic acid, and it is the highest in the case of sorba, and you can see here that it is enhanced 7 times in comparison to that of the ribosol. So these are the intrinsic dissolution rate, this is the equilibrium solubility, and this is the absorption coefficient. And here you can see that for the malonic acid, the solubility is the highest for the co-crystal with malonic acid, but for sorba, for SBRA, and for one form of adipic acid, which was the more dominant form, the solubility is enhanced in comparison to that of the drug ribosol. So this tells us that co-crystals are very interesting entities and have interesting applications in the drug industry. So with this, we come to a closure of this lecture, and now we will go on to the next set of topics from next week. Thank you.